The symposium. I'm glad you decided to join us today. I attend REU University, and I was really excited to get the opportunity to study at the Sevilleta this summer. And so I welcome you to my presentation on the effects of drought exposure on below-ground productivity, fruits, and the desert prairie. So first off, let's talk a little bit about environmental health. As many of the REUs know, I'm extremely interested in environmental health because I'm in a master's program, a master's dual degree program with the School of Public Health at REU University, and I am in a specific dual degree track where I'm studying environmental health and how ecosystem processes relate to humans. And so, in my definition of environmental health, it's promoting a healthy environment and healthy people. This can be implemented through various measures such as epidemiology, occupational hazards awareness, pollution remediation, and conservation measures with impending climate change. So, what led me to this ed? Well, first off, like I said, I'm in the master's dual degree program, which is interested in environmental health, and I wanted to see the connection of ecosystem processes and humans. I was interested in the LTER's mission in understanding these ecosystem processes, and I came to the Sevilleta to study and research ecosystem resiliency to reduced rainfall. So, I decided to study at the Extreme Drought and Grassland Experiment, known as EDGE. I studied under Scott Collins, who's my mentor, who cannot be here today because he's at ESA, doing amazing and wonderful things. And so I have two study sites, the blue and the black Rama grass-dominated sites, which is the Budalua gracilis and the Budalua aeropoda, respectively. And at these sites, they have different, they're different ecosystems, and there are various rainfall manipulation structures. As you can see here in the diagram, they look kind of like greenhouses. They're a little not pleasing to the eye when you're driving around the refuge. You know, you're like, oh, pronghorns, beautiful lizards, greenhouse shelters. What are these for? These giant gallons. Well, first I have three treatments. I have the 100% reduced rainfall treatment. I have the 66 reduced rainfall treatment, and then I have the ambient rainfall, which is the normal amount of rainfall that you would get during the monsoon season. And so there are various models across the U.S. They go in a gradient towards the northern plains. And in this picture right here, I believe it's in the Kansas site. And sometimes my mentor, Scott Collins, will visit there and additional sites to be able to check and see how the processes are working. And so at these sites, we test for species composition and above ground productivity and below ground productivity, which is my specific area of focus. So why roots? Why am I here? Why do I study these tiny, somewhat insignificant little pieces of grass or little pieces of plant? Well, first off, they're incredibly important and imperative to water transport within the plant. Here in the desert southwest, these plants are xeric plants, which means they're accustomed to arid, dry climates, and they have shallow lateral spread to be able to absorb as much water as possible in rare rainfall events. And so that's why it's incredibly important to understand how these roots work in order to be able to see the productivity of the grasses. So relating this back to environmental health. Well, first off, there's a huge demand of arable and usable agricultural land here in the desert southwest. It's important, these the Budalua black grasses are important for grazing cattle, and also root health is important for plant use for humans. And so understanding how healthy roots can support a healthy ecosystem is imperative to my study. So my research objective, well, I wanted to observe how root productivity can change under various drought conditions and various treatments throughout the growing season here. So I implemented these little root ingrowth bags, which are 20 centimeter cylindrical mesh bags that allow for roots to be able to grow in them in different and various angles. And now I have 8 at each site, and I implemented these root ingrowth cores. Then I later extracted them, and then I analyzed them in the lab for the percent biomass of each root ingrowth core. So I implemented these root ingrowth cores. I implemented 240 in the destructive sampling subplots at my site. Since these rain out manipulation structures, I have like my own little section. Scott has his, and other researchers have them as well. So I have my own little section where I could do some, root ingrowth implementation. And I'm also planning on collecting 180 standing crop samples. However, I have only collected 90 cores in and 58 standing crop samples thus far. So this is definitely going to be a continuation of a project later into October and in the fall. And I will be continuing this at my home institution at REU University. And I dug these root and growth cores approximately 15 centimeters to be able to capture the majority of the soil profile. And so I sifted soil and implemented them inside root and growth cores and vegetated clumps at the plot. And also, I don't know if you can see, but this person is putting in work in terms of muscle. And so over the summer, in order to be able to get that deep, you start hitting a cement-like layer. And so you kind of have to be muscular to be able to put them in. And so I definitely gained some muscle mass. So being a field ecologist, it's not only using your brain, but being able to use the field equipment. And so you kind of have to be an active person to be able to do the kind of work I did over the summer. I don't know, the people who have come out to help me, you'll nod and understand that there was some hard work to be done. Hard work. And so I later returned to the lab with my little roots and my little soil my little soil samples, and I collected and sorted them out with one of my tweezers, yes, the one that, my tweezer. And so I collected my roots, but methodically going roots, okay, not a root, stem. And so I did that, and that took a little bit of a while. And then I also dried them at 68 degrees Celsius in one of these drying ovens. When you get them back out, it's kind of like a batch of cookies. You're like, yes, oh, they're cooked, they're ready. And so that extracts out the water. That way I can just isolate the root mass. And then I also weighed and compared them to the total mass of the soil collection I had. And so my results so far, these are inconclusive because as we saw with the numbers I had before, I have to collect over 400 samples and analyze those. And thus far I have only 150-ish. So if I give you preliminary results, I don't want you to have the wrong idea and see things as significant that aren't. So I'm waiting to give you those results later on, but I'm just showing you that I've looked at things already, kind of. And this is a jump database, so I was just implementing things in here, kind of like an Excel spreadsheet, but cooler. And so I just want to discuss some potential bias that I may have already encountered. First off, this is preliminary data. I don't want to give you the wrong impression, like I said. I also see that root core angle, the implementation of when I put the root cores in, can sometimes have effects on the root growth. As you can see in this diagram, roots can grow different ways, but the majority of them, when you have them straight up, will grow this way. And so just the angle of implementation can sometimes be a variable in the amount of root productivity you can get, according to Daniel Makunis, I don't know if it's that in one of my literature pieces, but that's a potential bias I may have encountered when I implemented the root cores. In addition, root turnover and dead biomass is a huge thing. Since decomposition out here is kind of rare, dead mass can still be in the ground, and so that may be oh, I'm collecting biomass, but it's really actually dead, and it's not actually been isolated in the bag, and so root turnover is a huge problem, I guess out here, not a problem, but a factor, and I'm going to talk about that a little bit later on. And then also too, vegetation cover varies at each of these plots, at each of these sites, and that may be a factor in where I put the roots. And so the goals, just to reiterate why I came out here, I wanted to understand the implications of climate change and reduce precipitation. I wanted to understand the ecosystem's physiological processes, specifically related to water use, and I wanted to understand our interactions as humans with the environment. My recommendations for future REUs are to look into microbial interactions, to understand the biochemical pathways, to see more factors of nutrient cycling, and then also to implement many rhizotron cameras, which isolate root turnover, and they take little snapshots of the root growth. And so hopefully we can get those at an edge. Obviously everything costs money, so that could be a long-term thing, but these would be excellent in isolating root turnover and actually seeing whether or not I'm taking accurate results or getting accurate data, so to speak. So my acknowledgments are to my mentor, Scott Collins, who, like I said, could not be here today, but excellent guy, Hawaiian shirts, chacos, really cool guy. I also want to thank Amersfoort for picking me for the REU experience here. It's been awesome. And I want to thank the National Science Foundation, University of New Mexico, Jewish Fish and Wildlife, and my fellow REUs.